Hello everyone. So in this lecture we're going to be talking about DNA, in this case the function of DNA in the cell, and the cell cycle. Okay. So again, this you have been shown this before. Again, this is the structure of ribonucleic acid RNA. This is the structure of deoxyribonucleic acid DNA. Make sure that you understand the differences between this. Okay. So again, DNA is double-stranded, RNA is single-stranded. In RNA, if you look at this tail here, you see an OH. That's called ribonucleic acid. In DNA, if you look here, there is no OH. Okay, that's why it's called deoxyribonucleic acid. Deoxy meaning it has no oxygen at this position. Okay, in DNA you have ATGC, A and T hydrogen bonds together, G and C hydrogen bonds together. In RNA, folks, we do not have thymine. We don't have T. So instead of T, we actually have uracil, U. So this is highly unique to RNA. All right. Now, these two nucleic acids are related to each other. Okay, and how is that related to each other? So maybe I should give you a brief background of the function of DNA. DNA codes for all the proteins in your body. So all the proteins in your body, growth hormone is a protein, insulin is a protein, okay? Collagen, which makes your skin look fantastic, is a protein, okay? All of those are coded for in the DNA, okay? So how do you make protein? How do you actually make protein? Well, you make protein by the process of transcription and translation. So here we go. So again, the DNA is obviously in the nucleus. So when your body needs a certain protein, that region of the DNA that codes for that protein, let's say collagen, opens up. Okay? And in goes mRNA, messenger RNA, and copies, in this case, the code for collagen. Okay? And then mRNA then exits the nucleus, goes into the cytoplasm, into ribosomes. And FYI, ribosomes are actually made up of a different type of RNA called rRNA, ribosomal RNA. And it's the factory in which proteins are actually made. And guess who the workers are, who actually, in this case, then create polymers of this collagen protein? Well, the workers are called a third type of RNA, and those are called transfer RNA. Okay? So if you want to think of this as a different analogy, the analogy is this is the Ford headquarters. Okay? You need to make some Ford Fusion, so therefore you get a copy of the Ford Fusion design. That copy of the Ford Fusion design then goes towards the Ford plant in, I don't know, let's make a plant in Sheboygan, Wisconsin. And then the workers, the unionized workers, the United Auto Workers of Sheboygan then makes the protein. All right. Note that the DNA remains in the nucleus, it doesn't exit, and that is one of the reasons why something has to enter the nucleus and make a copy, because this nuclear membrane prevents DNA from leaking out. Why is DNA leaking out very, very important? When cells get too old and their nuclei becomes leaky and DNA starts leaking out, that's normally the sign of death. That normally means that cell is too old, too degraded, too damaged, and your body actually gets rid of it. Okay? So I hope that kind of tells you how important DNA is. All right, so DNA, folks, as you can see, so DNA is made up into 23 pairs of chromosome in the human body, right? So the 23rd chromosome is the XY chromosome if you're male, XX chromosome if you're female, right? And they are actually arranged in the specific structures. They actually wrap around proteins called histones. Okay? Think about them as like if you ever had a thread, a spool of thread, the little circular thing in which the thread actually gets rolled around is actually the histone. Or toilet paper. The little brown round thing is actually the histone, and the toilet paper in this case would be the DNA. Okay, now why is this important? Before I mention that cell division, cell growth cell division, specifically speaking. It's not important for life. However, you need to understand, and again, I'm going to kill skin cells to death here, literally and figuratively. There are specific cells in our body that constantly gets abused and constantly die. Okay, skin cells is one of them. All right, you've been bumping into all kinds of things. Wind and this weather is kind of like completely demolishing your skin cells. Okay, you go and use loofah. Guess what? You're completely destroying your skin cells. Okay, well, we need to replace those dead cells, and the way that we replace those dead cells is this thing called the cell cycle. So there are two distinctive phases of the cell cycle. The interphase, which is this blue here, and the mitotic phase, or called the M phase. Okay. Now the 23 and 1 hour, folks, is just relative to each other. Basically, what this tells you is that majority of the time, a cell will always be in interphase, and a very, very small portion of the time, it will be in the M phase. Okay. There are 3 subphases inside interphase. Interphase means this cell is just chugging along and it's just doing normal stuff. Normal stuff, it's in this case responding to stimulus, right? Maintaining a stable environment, creating energy. Okay? So G phase is basically what you're doing is you're getting ready to make extra DNA. And this is actually a very, very important thing to do for a cell to do before it divides. Human cells have 46 chromosomes, okay, or 23 pairs, right? So I need to start, like, it's like building a house. You need to get like the loader ready, you need to get dirt ready, you need to have wood ready, okay? But then in the S phase, this is where DNA gets duplicated or replicated. So before you had 46 individual chromosomes. In the S phase, after the S phase, we need to have 92 chromosomes. All right? 46, you go to 92. You double up your DNA. All right? So then, there's checkpoints inside each of this. So there's a reason why there are 3 distinct phases, is that every point of the way, if there's something wrong, this cell cycle does not continue. Okay, and then the G2 phase is, again, is making sure that everything else is okay. And the very important thing, folks, is centrioles. Centrioles need to be duplicated as well in the interphase, and centrioles are the ones that actually pull on DNA, right? And then what happens then, folks, is after interphase gets completed, you have 92 chromosomes, you have 2 centrioles, everything is correct, there's no leaky DNA, you have enough energy. Then you can proceed to the mitotic phase. Now, the M or the M phase is called mitosis, is called the division of the nucleus. That means you are dividing DNA. You have 92 here, you're dividing them into 2 cells, each one have 46 chromosomes. Well, the first thing you need to do then is you need to, in this case, remove the nuclear membrane. Because otherwise, if the nuclear membrane is still intact, the DNA cannot divide, cannot go into separate poles. So that's the first phase, prophase. So the DNA, the nuclear membrane evaporates, okay, gets degraded, and then the DNA becomes visible into the chromatin structure, to the chromosomal structure. And then metaphase, they line up in the middle. Why do they line up in the middle? Well, folks, before you had 1 chromosomes. Now you have 2 1 chromosomes. So in this case, the reason why is because you want the 1 chromosomes to be line up in the middle. Okay? And the reason why they have to line up in the middle is when anaphase starts to happen, in which the mitotic spindle from the centriole starts pulling on DNA, you want each pole to receive a copy of the chromosome. So each side needs to have 1 chromosome, the first chromosome, the second chromosome, the third pair of chromosomes, so on and so forth. Okay? And then telophase is when you actually then, in this case, have all the DNA into 2 separate poles, and the cell starts to pinch off or starts to divide. Okay? And cytokinesis, folks, and in telophase, actually the hallmark of telophase is the creation of two nuclei. So at this point, you're going to have a cell that has two nuclei and looks like a dumbbell. Okay? And then you have cytokinesis. Cytokinesis is just the splitting, the physical splitting apart of two daughter cells. Very, very important here. Both cells must be identical to each other, and they must be identical to the parent cell, in this case, the original cell. This is actually how, okay, cancers actually occur, or mutations occur. This is when you have aberrant daughter cells. Okay? Here's a further explanation of the M phase. 2n, n is, folks, is 23, so 2 times 23 is 46, right? You want to make sure that, again, at this point, you actually have 92 chromosomes before you actually proceed through the prophase, so on and so forth. And at the end, you can see this as 2n. At the end, the two daughter cells also have identical number of chromosomes. Okay? Further explanation of metaphase, they line up in the middle. Why do they line up in the middle? Look what happens in anaphase, folks. Each side receives a red chromosome, or the first chromosome. Each side receives the second chromosome, or number 2 chromosome, or number 21 chromosome, or number 19 chromosome. Okay, you don't want just one cell getting both. You want both cells getting one copy each. Folks, that is it.